So we're going to now begin. If we could have the first speaker, who is known to all of us and loved by many of us, and that includes Marty Murphy. Amy Abernethy needs no introduction. If you start, it's going to take almost all of her time. Amy, we're delighted that you're going to lead us off.
Thank you very much, Marty. Hi, it's delightful to see you today. My name's Amy Abernethy. I'm the Chief Medical and Chief Scientific Officer at Flatiron Health in New York City. I'm a member of the National Cancer Policy Forum, and today I'm going to talk about what can happen in the landscape of data sharing as we think about data sharing, we think about real-world evidence, and how this might support the development of IO combination therapy and this overall landscape. I am Amy and Mia. So Mia Levy, and it's very hard to fill her shoes. She cannot be with us here this morning. So I have added in remarks about the mechanics of data sharing as well as a part of the next 30 minutes or so. Fantastic. So the way that I thought I would address this is first give you a few taste tests, some case examples of what we're seeing mostly in the IO landscape and other places so that you can start to see how aggregated real-world data might help to serve as a solution point. We'll talk a little bit about mechanics and sort of aspects of data sharing, and then I'll explore this kind of question of pre-competitive collaboration because I think it's a really interesting question, and then some policy implications. So I submit to you that real-world data offers another kind of discovery. So works in concert with our traditional clinical trials and our traditional research mechanisms. What you're looking at here is a cohort of lung cancer patients. These are patients who've had their data from their electronic health records fully curated. And this overall cohort of lung cancer patients have very specific criteria for how they're selected. So essentially have eligibility criteria to get into the cohort. Each individual patient is receiving additional care, additional follow-up and observation across time. This is coming in through electronic health records, so the doctors who are using those EHRs have more patients. So as those doctors see more patients that meet the eligibility criteria, the cohort itself grows. And so it's a growing cohort that's being continuously curated and aggregated each month. And what you're looking at is data through June of 2018. So this is the end of June of 2018. At that point in time, there were 48,000 patients in this cohort. So I think the last time I presented this cohort in this room was about 2 years ago, and there were about 20,000 patients, and the majority of them are coming from the community oncology setting. Things that you see. So over here on the left, what you can see is that a quarter of patients have squamous cell histology. Over on the right, what you see is 12% of patients are non-smokers. What's interesting to me is I just sort of do a general cross-check every single month and watch these numbers, and these have been stable since I've been watching this data set for 3.5 years now or so. Smoking is an interesting one because we would expect that smoking was a structured data point in the electronic health record because it was actually required through meaningful use. Turns out it's required through meaningful use, but people type it in as in non-smoker, has never smoked, 1 pack per day, and it's still not something that you can easily analyze. And squamous cell is something that's coming out of the pathology report. So the pathology report is text and now needs to be translated into something that we can count. Second thing you're looking at here is the uptake of PD-L1 biomarker testing in advanced non-small cell lung cancer. And here we are over here on the left in January 2015. Here's June of 2018 over here on the right. And what you can see is the uptake of PD-L1 testing in the US. And you see that it happens in concert with critical FDA approvals. Most notably here is that what you're seeing in October of 2016 is the Keytruda approval for first-line and PD-L1 positive patients. And boom, when that approval happens, the rate of PD-L1 testing goes up in the United States. Now, as we're going to come to in a few minutes, PD-L1 testing, as we all know, there's lots of different ways that not only is this getting done in different assays, but also different kinds of reports and interpretations that are happening by the doctors. So coming up with one data model to make sense of this and see this at scale requires actually thinking about how to put this information together at scale. Now what you're looking at is a histogram. What you're looking at is a histogram across time. So this is Q3 of 2014 over here on the left, and this is Q2 of 2018 over here on the right. And what these are, each bar represents the different therapies being received by lung cancer patients across the US, so in this cohort. And importantly, the yellow is as the PD-L1 inhibitors start getting used. Now, a couple things to note here. There's very little PD-L1 usage in Q3 2014. When we drill down into the charts, what we can see is these are patients predominantly on clinical trials, compassionate use. And out here, what we see is that nearly half of patients are receiving PD-1 inhibitors. Maybe we're not seeing, oh, there we're seeing it again. And then the other thing that you notice is that this reflects markedly changing standard of care across time. If we're planning our clinical trials, if we're thinking about what our comparator arms are, the comparator is changing across this period, and we have to think about that. Now what you're looking at is second-line therapy. So the second-line lung cancer therapy, and what you see is that, in fact, PD-1 inhibitors popped up right here, Q2 of 2015. They become about half of all treatments, and that's stable across time. And what you see here is first-line therapy. In line with those approvals, there were some patients that were getting treated before the approvals actually happened. And when we look into the charts to understand what's going on, these are physicians who are making decisions together with patients, sometimes on trial, sometimes off-label. And in concert with the label changes, what you see is uptake across the US. Jimmy Carter. So as this story is unfolding, now in melanoma, we have a 91-year-old gentleman known to all of us who receives Keytruda. I think we all know this story, and we all know the price tag that was on CNBC attached to it. And certainly this got a lot of play. And what's been interesting is if we look in the medical records, we can see the conversations happening between doctors and patients about, this is what Jimmy Carter's getting. Can I have this for me? So what we're seeing is across the country, decisions are being made in the clinic that reflects the real world that we now can incorporate as its own and new type of science. This is work we did together with the FDA where we generated a data set that represented the first, it doesn't like me. Thanks. I'll tell you about the data set while Cindy's getting that. So where we just generated a data set representing all patients in the Flatiron database that had received PD-1 and PD-L1 inhibitors in the first year post-approval, and where we had at least 1 year of follow-up. And what you're looking at then is 1,344 patients. This is the first data set. There's another one that's coming out and was just delivered about a month ago that represents the next year of follow-up and all additional patients. And what you see here is that in the real world, despite the fact that on clinical trials, less than 8% of patients are 75 and older, in the real world, 27% of patients were 75 and older, and 64% of patients were over the age of 65. Remarkable difference from what we have in our clinical trials. However, if we look at the relationship between, for example, age and overall survival, in the left-hand panel, what we see is there's no relationship between age and overall survival. And if we look over here between line and therapy and overall survival, we see no relationship. Importantly, this was the first look after the first year of utilization of PD-1 and PD-L1 inhibitors in lung cancer. This story may change across time as doctors get better at selecting specific patients and as we get better at using these drugs in specific lines of therapy and in concert to the other drugs that these patients are receiving. So this is not a single static piece of information, but a piece of information that's going to be evolving over time. However, as you would expect, PD-L1 expression does predict survival, which helps you understand that indeed data sets are performing as you would expect as well. So what does this story really tell us? So first is why we're all here today. There is an evolving landscape with exploding pipelines, the demand for combinations, while at the same time a landscape of changing background. So as we're developing our drugs and studying new combinations, the background within which we're conducting those studies is changing as well. It's a time of more and more personalization, demanding of us to think about biomarkers while we're also simultaneously trying to make sense of cost. And we've got a changing policy and regulatory landscape, and within that landscape, trying to figure out how we take advantage of new policy frameworks in front of us, including 21st century cures. One of the things that 21st century cures offers us is the opportunity to just start to use real-world evidence and real-world data. So in fact, 21st century cures compels the FDA to develop a guidance and a point of view around real-world evidence, including real-world evidence potentially in support of label expansions and post-approval, satisfying post-approval requirements. So those are the two things specified in 21st century cures. This does not say that the FDA must accept it. It's that the FDA needs to take a look and provide guidance in how to think about it. This is the way that I've been, kind of framing up what I think is happening in my mind. So here's our current drug development paradigm, where we essentially go phase 1, 2, 3, 4, regulatory approval, and general uptake across the market. And instead, what we're seeing, especially as 21st century cure starts to come in, is a movement now towards increasing use of early approval mechanisms, followed by a goal of starting to monitor what happens as drugs get out into the general marketplace, and use of potentially real-world evidence to monitor that overall setting. However, if we're going to use data, that's passively being collected through systems and now going to be used in support of making decisions about how to take better care of patients, we better make sure that these are data that we can rely on, and importantly, that these data are adequately representative of the populations for whom we care. So now what I'm going to do is switch over and talk a little bit about what are real-world data. The FDA has on its website, and you'll hear more from Sean in a little bit, about what real-world data are and where they come from. Increasingly so, real-world data is often thought to be data that includes information that's in electronic health records, claims data, and other registries and types of information. Importantly, the idea here is to take advantage of all of the data that exists outside of the traditional clinical trial setting in support of now understanding how to better take care of patients. Real-world evidence, therefore, is evidence that we can clinically count on that has been generated from real-world data. However, if I am thinking about evidence that I can count on, there's a whole bunch of things that need to go into that. I need to understand how are the data sourced, how are the data cleaned and curated. I need to think about whether or not this is data of adequate quality that I can count on for the question at hand, and then the analysis of the question at hand. So the study being conducted with these data needs to follow a prescribed set of analytic requirements in order to make sure that this is done in a consistent and high quality way. Some contemporary features of real-world data that I think we're going to be seeing more of include that it's aggregated at scale. As Marty was talking about Project DataSphere, what you saw was over 100,000 patient lives represented in the overall data set, aggregated at scale. Another thing that you see is that these data sets, especially in the real-world setting, more and more are cleaned and curated in very detailed ways. You should be always able to get the specifications of how that work is done. A common data model is critical if you're going to have rapid analyses on the other side. So there needs to be a common standard for how the data are coming together. Ideally, multiple data sets all have common data models as shared data models between them, so they are rapidly linkable, because linkability is another key feature of these data sets. So for example, I can take data from the electronic health record where I've got pre-specifications of information, including personal health information in the right settings, and link those data sets to other critical data sets in the real-world data setting. And you're going to see an example of that around the linking of genomics and electronic health record data from Graph in a few minutes. Ideally, in this setting of real-world data and real-world data sharing, they're readily analyzable, and I'm going to show you an example of that. And importantly, quality should be assessed. We tried to set out a framework for how we could think about what are the characteristics and the guidelines around real-world data in order to sort of document these are the things that you need to know about. And this was a potential checklist that we put together and surfaced last year, including these features that most of these data sets now have within their documentation. A couple of details, and I'm not going to spend a lot of time on how these data sets get generated, because I'd rather spend time on examples of what can be done in with these data sets in service of the questions at hand. Importantly, as I mentioned before, you need to think about whether or not the data sets with which you're working represent the populations of interest to you. Many of these data sets are being sourced from across the US or even internationally. This is an example where Flatiron data are being sourced, and our data are coming from electronic health records, coming from lots of different clinics, over 250 different cancer practices across the United States. Another question is whether or not the data elements that you're interested in are in the data sets of interest. And this is an example of one of the data challenges to be solved, which is unstructured data that exists predominantly in PDFs. And more solutions are coming into play to help to solve for this. At Flatiron, we use a data abstraction process that we call technology-enabled abstraction. Other organizations are using more and more machine learning and natural language processing. At Project Datasphere, these data have been curated previously as a part of clinical trials data sets, so there's different ways that this is being dealt with. In this example, which is a PDL1 report, what you see is many of the details that we need are embedded within the pictograph, really sort of the pictograph being the PDL1 report itself, which is a PDF, and they need to be pulled out and put into specific data elements that can be counted and analyzed. Data curation, as I mentioned, is a part of the endeavor. It's not just unstructured documents, but it's data curation of all types. It's actually making sure that all of the data is cleaned up and readily available as needed. And at Flatiron, we use a combination of people and technology to curate data, but there's a lot of ways this is getting solved out there. Data quality is a critical feature. I could talk for like the next 5 days about data quality, so I will try and avoid that. But one of the things I'd like to point out here is every single variable in these data sets have its own characteristic. That characteristic includes how likely is it to be complete, and that depends on source, as well as how likely is the data point itself likely to be reliable. And I'm going to show you a couple of examples of how this comes into play with mortality data in a few minutes. So rather than spend a lot of time on the mechanics of how these data get cleaned up, what I thought I would do is show you some example use cases in service of the topic at hand. So the first is in line with what Marty mentioned and what was discussed yesterday, which is the development of external comparator arms or external control arms. In this particular example that I'm going to show you, it is an example from Roche. I'm going to be very careful to mention who these examples are from because it's important to recognize that different groups are doing different things. So in this particular example, Roche's drug alectinib was the third to market ALK inhibitor. At this point in time, they had a single-arm trial, which had led to their early approval here in the United States. Ex-US, they were waiting for the randomized trial to secure reimbursement. That randomized trial was pending. It was called Ascend-2, and what they asked us to do at Flatiron was to essentially predict what was going to happen in the randomized trial, whereas alectinib randomized to ceritinib. So what we did was take the eligibility criteria for that trial, identify all patients in the Flatiron data set that met those eligibility criteria, and then handed that data set to Roche. And then what they did was do a propensity score match between their clinical trial data set from the single-arm study to our data set in order to simulate what they thought would happen in the randomized trial. So what you're looking at here in the top panel is the alectinib arm from the single-arm study. What you're looking at in the green is the Flatiron simulated control arm using real-world data. About a year later, the Ascend-2 trial did ultimately read out, and what you see here is the control arm from the Ascend-2 trial. So that was actually a year before the Ascend-2 trial read out. Importantly, as a part of this story, this allowed Roche then to take this information to 20 or more health authorities around the world in order to secure reimbursement before the randomized trial came forward. Now, I have emerging examples within Flatiron that are looking at immuno-oncology agents, including combination therapy, but I checked with my statisticians and they didn't feel like they were cross-checked enough to show you the slides today. So the next one I thought I would do is talk about assessing outcomes and safety in patients excluded from a trial. And this is another Roche example, and it's with Kadcyla, and some of you have already seen this. But this was actually to solve for a post-marketing commitment. In this particular case with Kadcyla, there was concern, appropriately so, about enrolling patients with low EF in their pivotal trial, and so therefore those patients were systematically excluded. However, there was then a post-marketing request from France to understand what happens in the real world when patients are exposed to Kadcyla in the setting of having low EF. Roche actually tried to identify these patients that were accidentally put on trial and with low EF, or patients who were in registries, and they found 3. And so then they came to us and said, can you help us meet this post-marketing commitment? We had a set of eligibility criteria that essentially took patients with breast cancer in our data sets, looked for all individuals who'd received Kadcyla, and then we went and looked in those charts to identify whether or not the patients had evidence of low ejection fraction, and it identified over 100 patients in the United States who'd received Kadcyla. This data set is now just reading out, but what you see here is we were able to come up with a common data model to make up for the fact that EF was being represented in many different ways in the charts. MUGAs, echoes, doctor's notes, et cetera. And so we developed a common data model that could be accepted as a post-marketing study. And then what we do is take a look at data in context. So what you're seeing here is what we call a patient journey, so our ability to actually take a look at data in context for each chart. Kadcyla was received in the second line, which is the light blue dots. That was where Kadcyla was received. And the blue dots below are ejection fraction. So what you see here is this patient received her Kadcyla with an EF of 45%, and we were able to monitor her ejection fraction across time. And then we were also able to look at corresponding other events. So in this particular case, one of the other things to look at was hospitalizations, and then to go and look at those hospitalizations to discern if they were cardiac or cancer related. So these data sets can now start to be used in service of answering a lot of these more detailed questions that we have, especially as we think about the role of IO and how these patients fare across time. And this then met the need of what was required in France. In this example, this is work that we're doing right now with the NCI, and we have a similar example with the FDA of looking at patients who've been excluded from a clinical trial and evaluating specifically patients who started off with impaired renal or hepatic function at the time of starting IO therapy. This particular example is for melanoma, which is what the NCI has been working on, and the FDA has been working on non-small cell lung cancer. So here, the question was whether or not patients with baseline renal and hepatic function would fare, how they would fare in terms of both efficacy of therapy as well as potential safety concerns. And this was a poster that was presented at ASCO a couple of weeks ago. In order to identify these patients, we went to the melanoma database and then identified all individuals who had at least one laboratory measure for hepatic or renal function within 30 days prior to the receipt of PD-1 therapy. And specifically for renal function, we looked at creatinine, and for hepatic function, we looked at AST, ALT, and T-bili. And we scored these patients using CTCAE criteria. And what you're looking at here is the renal data on the left and the hepatic data on the right. These are small data sets right now, and what you can see is that there were 37 patients identified who had grade 2 or above renal dysfunction, and 31 patients identified who had grade 2 or above hepatic dysfunction receiving IO in the setting of melanoma. If we look at the renal patients, what you can see is that the patients who've got renal dysfunction are older, and significantly so. that they receive drug for about the same period of time. So the median days of receiving therapy is the same, but there's distinctly different survival between these two groups. If we look at the hepatic patients, what we see is that these patients are generally in the same age group. So hepatic, and this makes sense given the fact that these are likely patients who have, for example, hepatic disease from their melanoma. What you see here is they receive drug for a shorter period of time, likely going along with the fact that these are probably patients who have hepatic metastases and are very sick, and their survival is very, very short compared to the patients who did not have hepatic dysfunction. Now what we can start to do is also look at how these patients fare across time, and this is work that's now just starting to happen, as well as was there any differences in, for example, dose of drug received, periods of time when they got additional toxicity, et cetera. And then the last example that I wanted to share with you is something that I think is the other part of how real-world data can help to serve the questions in front of us, which is that I submit to you that part of the task is having data sets ready to answer questions quickly. So in this next example, I need to say up front that this is still in what we would call code review and quality cross-check. So this is not intended to be take-home information that's already been submitted for public peer review. But what happened was at ASCO, I asked a colleague, can you tell me what the top 2 questions are that you think are most important to answer? One of the top 2 questions that he said to me was that I'd like to understand whether or not we should be treating post-progression with IO agents and lung cancer. I said, well, that seems like a pretty important question to try and answer. And so first thing we did when I got back to the office was we took a look at how likely it was for patients with lung cancer to be treated post-progression, given the fact that there are observational and other data suggesting that patients may survive longer when treated post-progression. And what we found was indeed about 50% of patients were being treated past the time of progression in lung cancer. And then what we looked at was there any relationship to survival. If we look at all patients who've been treated post-progression, there is no relationship. But when we start to look at cohorts of individuals who have been treated 30 days or more post-progression, there seems to be a survival relationship. Importantly, there's a lot of confounders here, right? So you're pre-selecting patients, et cetera. The goal here is to have data sets that are ready, as well as very clearly stated questions at hand and very clearly analytic plans, as well as one of the reasons I told you about both code review as well as cross-check, the importance of peer review and thinking about what the results mean. But you can start to answer lots of questions quickly. So in the last 5 minutes or so, I wanted to talk about the fact that more and more organizations are working on this. This is work that was presented at Friends of Cancer Research last week, where over the last 6 months, 6 different organizations, actually initially 10 and then down to 6, came together to try and answer the same question together in IO. So specifically, what we were asking was whether or not, essentially what was overall survival with the use of PD-1 and PD-L1 agents in advanced non-small cell lung cancer. Each of these organizations both participated in the development of the work plan, including cohort selection and the analysis plan and the endpoints, and then each of these organizations generated their own independent data sets and answered the questions at hand. What was interesting is that it took us 6 months really to do this, from sort of time of getting everybody on the phone together to being ready to present the data publicly. In the last 2 months were the analytic time. So this actually happened very fast. It was also quite remarkable that all these organizations that are public-private, that are selling data and selling other kinds of research, all agreed to work together because we saw this all as a common problem to solve together. The problem being agreeing on how do we specify endpoints, how do we come up with a common data model, how do we do this work. Data sets spanned the size from 269 patients to 6,924 patients. What you see here, and I'm just going to sort of show the Forensic Cancer Research slides because this is really FOCR's work, is that there were very common characteristics across these data sets in terms of age, histology identified, even gender, although data sets had different representation of line of therapy, as you see in this bottom right-hand corner, which represents the different timing of when these data sets were pulled and curated, and how that reflects the shifting paradigm that's happening across the IO landscape in lung cancer, like I showed you up front. What you see here is, interestingly, if we look at the relationship between age and overall survival, which is on the left, just like I showed you before, not a single one of these data sets saw any relationship between overall survival on IO for patients who are under or over 75. It was very similar to the results that we saw before. What you see here is the distribution of overall survival estimates from these 6 different data sets. Importantly, you're seeing something that's a feature of real-world data that's particularly important to think about. What happens when you have missing mortality data? People look like they're living longer. So what you're seeing here is some of these data sets have missing mortality data, which is something that they recognized in the mortality endpoint available to them. And so you see differences in overall survival that's likely a feature of the mortality data point, as opposed to whether or not these patients are specifically living longer or less long across these data sets. So importantly, one of the things to think about and understand is, how do we understand what are all the characteristics of the data set and how it can answer the question at hand. However, time to treatment discontinuation is a feature that's very consistent and very high quality within all of these data sets, and you see much more consistent findings across these data sets. And so this is time to treatment discontinuation. Importantly, to get people together, having this conversation and talking about what's it going to take for us to be able to do this work at scale and solve these problems was one of the most critical things that came out of this book of work. So what we saw was that there are lots of people who are interested in coming together to solve these problems. There is a similarity, despite the differences in the data source, that we don't have data standards for many things, such as PD-L1 testing. So only 2 of the data sources were able to present information about PD-L1 testing or disease progression. And we're now working on thinking about, can you harmonize? Can you pull these data sets together in some kind of way? So what if you tried to combine all those data sets? What would that take? So my question then becomes, do you do differing analyses and look at the same question from multiple points of view? Do you pull data together and try and have one data set to rule them all, so to speak? And there's different ways of looking at that. But this kind of question of, can you now start to pull these things together and have, for example, translation tables that allow you to harmonize when you've got different data models underneath the hood? Which takes me to sort of my last few comments, which are around the concept of strengthening research through data sharing. So I first submit to you that when we first started talking about data sharing, and I think I took this podium around this topic for the first time in 2008, at that time, I think I had the naive belief that if we could just dump it all in there together, we'd actually solve a whole bunch of things. And it turns out that if you just dump a whole bunch of data together, you dump a whole bunch of data together and you just get a mess, right? So that if we imagine that we're going to just submit to the New England Journal and send everybody our data sets without the specifications and ways that we think that we pull these data together and how we're going to do the cleaning, I submit to you that it actually doesn't lead to data sets that you can then analyze and make sense of in a confident way. That being said, data aggregation and thinking about data aggregation at scale, including the hard work of curation and understanding the details and documenting data quality and knowing how to analyze it and knowing what the results mean, actually does get you to a place where you actually can make sense of it and see new things. There are a number of data sharing consortiums that are happening out there, and I think all of them are experiencing some flavor of the same thing. Getting the data cleaned up are hard. This is an expensive endeavor often and takes time, money, and will. That there are differing data models, and we have to think about how to get those data models aligned and come together. And that ultimately, whatever collaborative it is, we learn critical things about how to do this work. And I would submit to you that the most important part of data sharing is sharing of the how. Sharing across the lines, here's how we think about this problem at hand. So I exposed one of our biggest problems in real-world data to you, mortality data. So how are we all going to think about solving that problem? Because it's one of the most critical data points that has to be solved in these data sets. Thinking about the how is going to be the most important part of data sharing. A few last sort of comments about data sharing. I think there's been a lot of belief that that data sharing is going to make things free. It's just not going to be free. I actually think that I thought in 2008 that it was going to be free. I think another thought about data sharing that we had at the time and that I had was that if we could just get it all together, then we'd find really cool algorithms to rule them all to get everything cleaned up. It turns out that as we build algorithms, including, for example, machine learning and natural language processing algorithms, those algorithms are specific to the underlying data set. And so the work to do all that cleaning and all that specification is actually real work that has to happen on an individual basis. When I see the language around pre-competitive collaboration as it relates to data sharing, I'm kind of like pre-competitive for what? I see how data sharing, especially across, for example, the pharmaceutical industry, can help as we bring clinical trials data sets together and we start doing this work. But I actually submit to you that having data sharing across a whole bunch of, for example, data vendor lines requires us to think about how do we keep those, essentially those vendors in business so that they keep generating the data sets that we're going to need in order to do this work. So we have to think about how do we do this smartly. I think the forensic cancer research example was a really great example of where we can find the benefit. We found a lot of benefit in the how do we do specific things and what are the problems to solve for things like mortality data. And then we need common data standards. We need standards for things like how do we come to a common data model for PD-L1 biomarker testing? How do we have common data models for things like endpoints, including disease progression and tumor response? The policy context is really in front of us. We've got enabling policy like 21st century cures. We have to think about privacy, security, and governance, but we already know that and we're all working on it. There are incentives in place for data sharing as we start to see how these data sets can be used to solve problems, but we need more and more incentives. We need mortality data. I could just stand here all day and say that. And then we need regulatory policy that helps to drive standards. And so if I make any policy recommendations, it's that standards will help the entire field. My take-home summary then is that these data offer us an opportunity to observe the interrelationship between diagnosis, treatment, outcomes in ways that we haven't been able to see before. They help us, in order to do this, we have to solve a lot of challenges, including things like data aggregation, curation, and assessment of data quality. We have to figure out this mortality data thing. I don't think I've said it enough. And data sharing isn't free. So thank you very much, and thank you for your time.